We need to get young people interested in chemistry from an early point, if chemistry is everything. And I think there's a chance to use the young kids' ability to see images and store these for later. We should therefore do animations, and this is something I always advertise for, trying to do animations of chemistry which are appealing to young people. Also for us, I think it's important to realize that when we do research with constraints, that is when we follow calls from funding agencies, we actually do research which is not necessarily what we really want to do. And also, when we make applications, these are made in a very short period of time before the deadline, and usually they would describe work that would carry out over the next couple of years, maybe 5 years. And therefore, I think that research should actually not be done by funding in applications, but rather by performance. So if we as researchers agree to have an evaluation that goes like 3-4 years back, and then that is what determines what we should how we should be funded for the next period. I think that would be a much more profitable way of doing funding for research. So I shared the Nobel Prize with Barry and Carolyn, two good friends of mine, and I'm very proud of that. But where did I get my interest for research from? And this shows a picture from my grandparents' forest. And I very early on got a interest in why things are like they are. What is it that causes that? And the answer to that, of course, is the chemistry behind it. So that was my first interest in chemistry, but the real interest came later on when I went down to Struer's company south of Copenhagen and bought 15 kilos of explosives to make things like what you see now on your screen. This was fantastic. I went back home, I remember, with a big cardboard box with these explosives, and down in my parents' basement made fantastic rockets and Roman lights and stuff like that, until I blew a huge hole in the lawn of my parents' garden, and then that was the end of that. But it spurred my interest, and later on I was so lucky to get to Carlsberg Laboratory, one of the finest laboratories in the world at the time. There I made an application to the Danish National Research Foundation for new organic quantitative chemical transformation reactions that could be used for combinatorial chemistry. And we would like to combine those with peptide diversity to do drug development, material science, and adding to STEM in the society. So we got the money, and we built a research platform. So mainly everything we did was based on these polymers, the PECA, SPOC, and POEP resins that we could make. And you can see here a picture of these resin beads, and they are very uniform, very well produced. As we could even do quantitative assays on these resins because they are swellable in enzyme buffers and organic solvents. So we could do a large array of organic reactions. We optimized those, and this is a list of organic reactions that we carried out and combined with peptides and assayed with enzymes of different kinds. So one of these reactions was the CuAAC reaction, and that is, of course, very important for later on. But we built this platform which starts with combinatorial synthesis, and we made these containers where we can do 20 individual reactions, mix all the polymer particles and do another 20 individual reactions. And that can continue around the cycle here to generate exponentially growing number of compounds. And they could then be assayed on these resins with enzymes like shown here. We have 6 beads that are enzymatically active. So this is a substrate for protease, and we collect that and analyze it, synthesize a protease. Here's it injected in a mouse, and you can see we can see this arthritis in one of the feet of the mouse, while there's nothing in the control, so that worked very well. We also synthesized catalysts, and what I will talk about today is this class of compounds, all sorts of very good pharmaceutical-looking candidates for by a new reaction called the INAIC reaction. We can also, by combinatorial chemistry, we found this peptide that can make us monolayers of cells as detectors of the compounds on the surface of the cell, of the beads. And he has shown a library of different compounds with one active. We isolate the active compound and we analyze it. We can do this by a strategy that is encoding of the individual beads with these microparticles that are distributed in the polymer. So this platform was really efficient, and we worked a lot with different types of chemistries. And one of the chemistries we investigated was the use of acetoacid chlorides as very reactive amino acid derivatives that could be used for synthesizing very difficult peptides like this one here. Can only be synthesized in this way, actually. And we thought this is a very good reaction. Maybe we can combine that with the acetylenes. So we made the copper acetylide here, and we coupled that with the acid chloride. This was Christian Thorne, who was a PhD student at the time. He worked at that for several months, and nothing came out of it. It was really, really difficult to get this to work. And at the end, he would like to maybe even quit his PhD study. But we looked at it, and it seems that he is forming only one single compound, and that was a triazole of the A side in the presence of the acid halide. So that was really interesting, and he then investigated the compatibility of this reaction with all the different amino acids in proteins. And in 2001, he presented this in San Diego. So what is a click reaction? It's a reaction with a very high enthalpy because we want it to be quantitative and we want it to happen at very low concentrations. We also need orthogonal click functionalities, which are orthogonal to the other chemistries that we have in our molecules. It should be easy to access and install, selective and biocompatible even at room temperature. It should be stereo and regioselective, and it should preferably be a triggered reaction, insensitive to carbon dioxide, water, and oxygen. And then it should be coordinative in the transition state, so it can assemble at low concentrations. Most importantly, it should provide something we cannot get in other ways, so provide inaccessible molecular architectures. But this is not very easy to find, and I'll come back to that. Here we are at one of the most recommended holidays in the world. It's in Stockholm, and this is the first day of a week that I was invited to. Very much recommended, actually. And here is a container that we used for our first combinatorial libraries, and that is now at the museum in Stockholm, the Nobel Museum. You also get to sign a chair here, and this is right beside Emmanuel Carpentier, so it couldn't be any better than that. We were looking at a lot of different organic reactions, and one of the most useful reactive groups in organic chemistry is the aldehydes. So we supplied our peptides with N-terminal aldehydes to do all sorts of additions of different nucleophiles and reactions like pitted Spengler what was amines and reductive aminations on these aldehydes. But we found that this only succeeded when we had methylation of the two penultimate nitrogens in the peptide. And this was work from Thomas Gross and Jörg Rademann, and we did an NMR study where we could see that we didn't actually have the aldehyde in the mixture. What we had was a N-hydroxylactam. So unexpectedly, the aldehyde would react with the two penultimate amides in the peptide. So we needed backbone inalkylation in order to get this to work. But it also opened up an opportunity, because if we have this N-acyl iminium ion formed, as you see here, the reaction occurred first to release an aldehyde. Here we release an aldehyde, it reacts with the amide, and then we have a hydroxylactam, which under acidic conditions will eliminate to create a very strong electrophile that can cyclize into this indole, and this is a Fidget-Spinkler type of cyclization, but with an N-acyl iminium ion. And now we wanted to know if this is a general reaction. The reason it happens is because the orientation of the aldehyde is actually very favorable for formation of this five-membered ring. We wanted to have some building blocks that could allow us to do this reaction in a versatile fashion to create many, many different architectures. And the first one we thought of was a malonic ester synthesis, where we first introduce the aldehyde. Then we do a protection of the aldehyde. We do another alkylation and the malonic position, and then we do a decarboxylation to get a building block in 50 to 80% yield here. So this worked quite well, and we could also do this with phenylacetic ester synthesis. It's exactly the same procedure, except we don't need the second alkylation because we already have introduced this aryl group. Now we could also do more in situ type of reactions. Here we have a diol that has been protected and oxidized to an aldehyde. We protect the aldehyde with the box protecting group, something we developed, very efficient actually for the subsequent reaction. We can then do the oxidation after cleavage of the TBDMS group. We can do the oxidation with tempo and sodium hypochlorite. And this gives us the carboxylic acid, and we can then make larger rings with these aldehydes. We can also use vinyl groups. So here is 2-vinyl benzamide and osmium tetroxide, sodium periodate, and DAPCO. THF was found to be extremely good conditions that works on solid support to produce an aldehyde from the alkene. And then from amino acids, we could reduce the amino acids to the aldehyde using the Weinreb amide procedures, and that gives us the aldehyde with the F-MOC group. We can protect the aldehyde with the box group, and then do the deprotection of the F-MOC group and the coupling with bis-pentafluorophenyl carbonate. That gives us now a reactive carbamate and an aldehyde with a optically active sidechain here. So a lot of different opportunities. When we do the reaction with different reactive C nucleophiles, that worked extremely well. As you can see from the HPLCs of the crude products released from the resin after this reaction. The first step here is a 10% TFA, and the second step is the cleavage reaction and neutralization of the compound in order to get the HPLCs. So We thought that is an interesting reaction, and we might actually look more into detail. Here you see the crude NMRs directly from the resin, and you can see two things. You can see that it's extremely clean, but you can also see that there is only one stereoisomer formed in these reactions, so they are completely selective for the trans orientation in these two positions. And that is independent on the nucleophile. We can also see what the absolute configuration is from an NOE of this, where we've used thiophene as a nucleophile. This is connected here, and you can see now we have a huge NOE between 1 and 3, and we have no NOE between 1 and 2 and 3 and 2. So can we do a lot of different things with this reaction? And the answer is yes. Here we have different substituted tryptophanes that you can buy either chirally or racemically. And we have our building blocks. Some of them are racemic. So in the next slide, I show you HPLCs where you will see two peaks, but these two peaks are all formed very cleanly. And this is because we have a racemic position here, so we get two peaks almost in equal amounts. We still have a optically active center over here, and then we have tryptophanes that cannot be obtained in a chiral manner. So these are also two peaks, but you can see extremely cleanly formed in this heterocyclic these heterocyclic compounds. When we did the activation, we found that it's actually important that you do first a weak acidic aqueous TFA activation to form the hydroxylactam. This can then be cleaved off, and you can see the hydroxylactam as two different compounds because there's a stereocenter. There's two stereocenters. And then we use strong acid conditions to affect the condensation. So that eliminates the water and makes a reaction happening to the pi electrons of the aryl part here. And we could make quite a lot of different pyrroloisoquinolines, as shown here in the next slide. We did a lot of different phenylalanines, now less reactive than the endols and thiophenes and so on. And you'll see that these are all quantitatively forming with a 50% TFA in dichloromethane, all quantitatively forming these pyrroloisoquinolines. Even when we have strong electron withdrawing groups like these dihalides, and this one here. So if we have substitutions that are electron donating, the reactivity of the aryl system becomes larger. And if you remember, there was only one peak when this was halide, but if it is this O-methyl group, you will see you form both the ortho and the para substitution to the methoxy group. This is even worse if we just take the hydroxyl, which is smaller than the methoxy. In this case, we get more of the ortho. But that is only when we have these electron donating groups. We can also use polyaromatic systems. So this is the 1 and 2 naphthylalanines, and they both react to give us these two substitution products. And here we are using pyrene, also forming the optically active substance in quantitative yields. See here, very nice products directly off from the resin. Here we are back in Stockholm, and this is a day of humiliation because you're giving a lecture for maybe 10 million people. I don't know how many looking at it, but it was actually quite difficult to get started. Once you get started, then it works fine. And we were all very happy when it was all over, here embracing each other, wishing each other congratulations with the result. Now we were also looking at heteroatom nucleophiles, because there's a lot of molecules that can come out of heteroatoms instead of the C nucleophiles. And you can see the yields are quite high, except for this situation. We don't know really why, but serine gave us always a complex mixture. Also, when we have an 8-membered ring on one side, it gave a complex mixture as well. When we do these reactions, we could form 5, 6, 5, 5 or 5, 7-membered rings. We could even use amides as nucleophiles for these reactions. When we didn't put a nucleophile in the sidechain, what happens is that the N-acyl iminium ion actually finds the next amide down the peptide chain and form these 2 5-membered rings. Here we again have very high purity of all the products. And what we're doing here is we are doing a Suzuki reaction prior to the cyclization. So we can do all sorts of reaction in the presence of this moiety. So these are the conditions that were optimized here to give quantitative Suzuki reactions on these 2 positions. And these are the yields, and these are the chromatograms. And you can see we can install any kind of moiety here as we like. So a very good reaction in general. And this was all work by Thomas Nielsen and Sebastian Lequemont. Sebastian also did this oxidation of the diols, first to the aldehyde and to the acid, and coupled it in to see if we could get different ring sizes. That is possible, except if N is 2 here, so that's a 7-membered ring and a 6-membered ring, then we didn't get any yield. And these reactions are all like on-off. Either it works or it doesn't work. And it's like 100% doesn't work then. So here are the results of some of these, and you can see 6 6-membered rings works well. We get almost 1 compound in all cases. And we could also use vinyl groups, as I said earlier, vinyl groups, and oxidize these with osmium tetroxide, DAPCO sodium periodate, and THF and water to give us the oxidation product that then react with the amide. That is what we see here. And then we could also do that with, we have 8 examples where we also do the INAIC reaction. And this is a result. You can see many of these you have seen before, but we can do these interesting molecules in very high yields. Besides the vinyl group I showed before, we can use vinyl benzamides to generate the aldehyde here, and then we can introduce 2 aryl groups around this heterocycle. And that works very well. You can also use heteronucleophiles here with threonine reacting with the N-acyl iminium ion, giving us this product in 95% yield. We can also use acrylamides. So if we have a proline or substituted amino acid here, the cis configuration is present, and that gives us the opportunity to make the aldehyde amide condensation here, and then the reaction with a C nucleophile to give us these compounds. And you can see it's not quite as clean as the other reactions, but it does give us mainly the right product as shown here. So we also try to use then, once we had established that, the allyl glycine as a source of the aldehyde. By oxidation with osmium tetroxide and sodium periodate, we can get the aldehyde, and that then cyclize in this manner. And we use this reaction to demonstrate that we can actually do this at the end of a peptide synthesis rather than as an initial reaction. So here we have a compound with a allyl glycine that reacts after oxidation with the amide, and then we can continue the peptide synthesis and synthesize this highly constrained agonist for the mu receptor. So this is the way that you would normally do this, but we also, as I said, try to do this on the intact peptide. So here we have synthesized the entire peptide first, and then we do the cyclization with the same yield as we get from the stepwise reaction. And this is what it looks like, very active on the mu receptor. Finally, we use the amino acid-derived building blocks to install urea. Instead of an amide bond, we have now a urea bond that binds the aldehyde to the peptide that we have grown here. And then we can free the aldehyde, and it will react, in this case, to form a 5-membered ring. So with the second nitrogen, and then with a C nucleophile to give us this type of compound. That worked very well for a number of different substituents, and you can see it's not quite as clean as the INAIC reaction. We call this the INAIC reaction. This is not quite as clean as the INAIC reaction, but it works quite well in most cases. When we do this reaction with aryl groups on both sides, so here we're using instead of the vinyl amide, we're using an aldehyde and a pentafluorophenyl carbonate. We react that with the N-terminal to get this product. And this product is nicely formed, but it's not stable in the presence of air. It will slowly oxidize to give you an intense red color. And we can do that by DDQ to get the oxidation to these products here. And the interesting part about these products is that they are highly fluorescent. And the fluorescence seems to depend on the environment very much. So we can here install in situ fluorescent probes in peptides, which is, of course, something you would like to do, in particular since the fluorescent probes that you can buy are extremely expensive. And we have a large variety of different fluorescence properties from these compounds. We can also do two subsequent reactions with these building blocks. So this is a pentafluorophenyl carbamate that is reacted with the sidechain here of this amino acid, and that forms now the imidazolone here on the nitrogen, and then we can deprotect the F-MOC group, take it off here, and then come in with a second building block shown here. And this building block attaches itself to the nitrogen, and then it forms a aldehyde that reacts with that nitrogen and this as a nucleophile. So here we can form a large variety of new heterocycles with different substituents, and all of this is very amiable at combinatorial chemistry, just showing you the purity of this particular compound after the cleavage of the crude product or from the salt support. So we have a reaction where we can form 5, 6, and 7-membered rings and 5 and 6-membered rings here. We have 100% trans stereochemistry here. we have the possibility to vary the substituents here, here, and here as much as we like. So that is a very rich class of compound, including that we have the possibility of taking an L, making the trans to the L, and then another optical activity down here. So we have LL, LD, DL, and DD. So we have a huge space of conformations. And these are the sort of a listing of some of the compounds that we have made. You can see the scaffolds are quite different from compound to compound. They look like this in 3D. And you can actually make this, and then you can see that we have really a huge goodie bag for pharmaceutical industry to address receptor binding sites of different pharmaceutically interesting proteins. Here we are back in Stockholm, and this is a ceremonial day. You get the medal from the king, and you thank everybody, and then the king just takes the medal out of your hands again, and you're wondering what is going on. You didn't deserve that medal or what. But this is because a lot of the Nobel laureates are very old like me, and they sometimes forgot the Nobel medal in the ceremonial hall. So nowadays you just get the medal on the last day, and then it's your own responsibility to bring it back home. We did a combinatorial assay where we isolated D-amino acid peptides, that were from this library shown here in the corner. And this is a superior compound for cell adhesion that we isolated from a very large library of 55,000 D-amino acids. This peptide sticks to the cell surface without harming the cell. And you can see here how we can grow now cells on the surface of the beads. The beads can contain this adhesion peptide, but also an activating ligand that can activate cells, as shown in this small slide up here. And then we can sort everything as we want in our bead sorter. If we activate the receptor in the cell surface and we incorporate here a yellow fluorescent protein in the expression that is activated by the receptor, then we can get fluorescent cells, and that's what you see over here, the control and the cells. In this case, it's melanocortin 4 receptor and alpha MSH that we add, and then you see the cells are fluorescent. We do this as single vector assay, so we have a single vector that both express the EYFP and the GCOP protein receptor. That works very well, and we can actually make quite stable cell lines with this method. So we express the receptor in the cell surface, and we bind our ligands to the receptor, and we get the effect on the inside of the cell. So we would like to look at this melanocortin 4 receptor and see if we could establish selectivity over the other receptors. These are click cyclized ligands, and here you see the assay, what it looks like. And this here is the most selective agonist for the melanocortin 4 receptor, which is not active with the melanocortin 3 and 5 receptor, or at least 300-fold less active. So we were able to make a compound that was selective for the melanocortin 4 receptor, and it's shown down here with the HFRW as the sort of main pharmacophore. Now we would like to also make our molecules to interact with those cells, and this is from this library I showed earlier, where we have one hit which really activates the cells. And then we will do the fluorescence analysis, and we will do the analysis on MS of this compound on the bead, and then we can find the actives. So we made a library with the adhesion molecules on the surface, and then with these compounds for activations of the so the 34,500 compounds for activation of the melanocortin receptor to see if we can make small molecules that activate this receptor. And these are some of the active beads that we picked out and analyzed, and the structures are on there. You will notice that most of these structures are tryptophanes, which is also what we expect. And these are the most active ones, and you can see they are very similar. Here we have the tryptophan, we have a benzyl, here we have a phenyl, and again a tryptophan, a phenyl, and a tryptophan. And this here is with an EC50, which is actually sub-micromolar. So that was quite interesting that we could take this small ligand and mimic the interaction with the larger ligand that is sort of a real life from the endogenous melanocortin receptor ligand-derived peptide. And this is how it binds, shown comparison of the INAIC ligand with the peptide, the cyclic peptide. So we were very interested in the CB1, CB2 receptor system because the CB1 seems to be the only receptor that really is capable of relieving chronic pain. The morphine receptors are good in the initial part, but they are desensitized with time, so chronic pain cannot be treated with morphine for a longer time. Here we have a synthesis where we start by putting on a phthalimido group on an amino acid, and we make the amide. Then we can reduce one of the two carbonyl groups with this reagent to the hydroxylactam, eliminate and react with the aryl system. So we generate this very reactive electrophile that interacts with the pi electrons and make a new bond here. So we have this compound, and now we are installing sort of a ligand that fits into the binding site on this bromide that's here. And now we have a compound that looks very much like tetrahydrocannabinol in the sense that it fits exactly, almost exactly, into the binding pocket. So if we remove THC from the pocket and put this in, you can see how well it is situated there. So what we want to do with this is, as I said, to avoid the psychotropic effect and just get the pain relief. So we look at the dimerization with 5-HT2A, which is the serotonin receptor that is also involved when we are, taking drugs to get psychotropic effects, because these dimerize with the CB1 receptor and are affected by the binding of CB1 ligands to give us this psychotropic effect. So if we can remove the dimerization, we can remove that effect. So we have modeled the receptor. So this is a HT2A dimer, which is a crystal structure, and we have modeled on the C and N terminal ends of this. Here we have the CB1 receptor, and that is also with the C and N terminal ends. And what we found is that the C and terminal ends that are sort of absent in the crystal structure or not included in the crystal structures, they are actually also important for the dimerization. But Matsukas and his colleagues have made a small peptide which is part of the CB1 receptor. It's this dimerization site, and they can actually completely inhibit the psychotropic effect of THC by adding this peptide, which prevent the dimerization. So we are in the process of synthesizing the peptide that does this, but also new ligands for the CB1 that should be able to prevent the dimerization as well. So I would like to end this with saying that we have 3 well-known click reactions: CuAAC, spark, and TCO reaction. There might be a few other reactions that you can mention, but I would like to add a delta S-driven reaction to the click reactions, because we have all the click criteria actually is included in this. And we see here the reaction to a quantitative formation of a stereochemically pure product. And we call this the INAIC reaction or INSIC reaction, depending on this part down here. So we are back in Stockholm, and this is the last day. This is the day where you go to the ceremonial dinner. And this is my wife. She is dressed up in the click dress. It's a very artistic, and you can see that because this is 6-membered rings made by the artists. And that should, of course, have been 5-membered rings. We haven't dared to tell them they are a mistake. But my wife saw this dress on the museum on the first day, and she said, you got to make me that for the dinner. And they said, yes, of course. And then they made it, and she took it on, and I was very proud to bring her to the party. But when you do that, you are in great danger. So this is the Prince of Sweden. And he quickly separated me from my wife. And then he took her under the arm. I said, oh, she took him under the arm. And they went, and he was seated between Philip and Daniel, the 2 princes of Sweden. And I was somewhere else in the room. So I was sort of a bit disappointed because she had dressed up that beautifully. But I got my revenge on the next day. This is, you can see how goofy I look. That's because this is, of course, Victoria, a very pleasant person from Sweden. And with that, I would like to end the lecture. So I would like to thank Claus and also Michael Boltz in particular for inviting me in here to this fantastic environment at Copenhagen University. Also, I'd like to thank Danish National Research Foundation, Carlsberg Laboratory, and the university, both for the facilities and for financing. And then I would like to thank my family, Fidwa, Gianni, Anna, and Sandra, for their support and so on. But mostly I have to thank all the co-workers involved in this INAIC chemistry, and also Christian for making the click reaction with me. Kasper, Frederik, Thomas, Sebastian, Daniel, Jörg, Thomas, Charlotte, Mads, and Jürgen are very important for this project. And with that, I would like to thank you all.